So now in data science, day 9, I'm going to continue the summary of the another book, that is Python based book for data science. And now the chapter is statistics. And this is really very interesting, where the author of the book has tried to bring the simplest form of statistics, which is most commonly used for data science. And in this case, as I was discussing about mean, median, variance, average, all such things have been covered very smoothly and in the easiest way. So how are you going to describe the single data set? For example, if you're writing the code in Python, MATLAB, R, or Perl, then you can see the sample here, how they are presenting the 5 values in one declaration. And this is quite obvious that you can write in this way. But if you are using R, it will be very close to this. In MATLAB, it will be without comma. If you want to use comma or semicolon or colon, it has different meaning in MATLAB. So you need to be aware about the syntaxes in different codings. So first thing is in a statistics part, that is histogram, where you can see in this plot, the histogram plot has been done for the number of friend counts. And this you can see easily that in the left side, that means in the beginning from 0 to 20, it appears to be number of people is quite high, but as you are growing up, then it is decreasing the number of friends to the number of people. So this is very easy to present or plot the histogram for such numbers. But this is one of the simplest picture of histogram. There could be different way of pictures, which brings some complexity in terms of coding. That is here, what you can see. After plotting the histogram, you can plot or bring a line over it in order to see the normal distribution or in which pattern the distribution is. Second way of presenting the histogram could be if you are presenting 2 or more than 2 data. So you can use 2 different colors for the same bar in different way, or more than 2 colors can also be used. That is why I have presented here different types of histograms with different presentations. This is another way. Looks really beautifully plotted histogram. That is probability of occurrence. And this represents the finest example of normal distribution for any data. And this is like what you can see in the middle. It is exactly showing 0, while in the left there is equal distribution and on the right side there is equal distribution. In general, we represent -1 to +1, and in the middle there is 0. But this is also the finest way of presenting the normal distribution by using histogram. Then this is another way of plotting the histogram for different kind of statistics. Then there is another next way of histogram patterns. For example, this is called symmetric distribution. Then right-skewed distribution. That means when you are going towards right side, then the histogram size is decreasing, and opposite to it, that is left-skewed distribution. So here it is decreasing, here it is increasing. So this is called right-skewed and left-skewed, and this is called purely normal distribution. Now, another term is coming, that is called central tendencies. So in all kind of distribution, if you want to see whether it is normal distribution or something else, then you need to go for what is the central tendency. Does it go for this kind of plot or something else? That is for another. So here this new term comes, that is called central tendency. So in central tendency, you are looking for mean, median, and mode. So here you can see, if you are looking for the mode, this is the mode in one type of plot. this is median, then this is mean. Another way of frequency plot, here you can see mean, median, and mode. For exact normal distribution, all the three lines fall in the same place. But when you are going for another way of the distribution, then mean, median, mode are shifting to other places. So next part is dispersion. Dispersion is also slightly different, what you can see. So dispersion means the distance of the scattered data from the mean or average value of the data, as you can see here. So measures of dispersion, what you can measure here in dispersion: variance, range, standard deviation, coefficients of variation. And this is the place that is called distance from the center. So next part is correlation. This I have discussed in the previous lecture. That is called positive correlation, negative correlation, and zero correlation. That means there is no correlation. For this purpose, you have some inbuilt functions in every language, every coding language. That is, for example, a Spearman correlation, Pearson correlation, and there are a number of correlation depending on your data. You have to choose which correlation will be more suitable for your purpose. So types of correlation, as you can see here: positive and negative correlation, linear and non-linear correlation, then simple and multiple and partial correlation. So in positive negative correlation, you can see the definition clearly defines when two variables move in the same direction, that is positive correlation, and the correlation value, that is rho, will be always positive value. Higher the positive value, and always the maximum value is +1. Higher the value, positive value, which starts from 0 to 1.0, that is the higher correlation value. And the same thing happens when you are discussing about negative correlation. Lower the value, that maximum, the lowest value should be -1. So from 0 to -1 direction, lower the value, higher the negative correlation value. Now it comes to linear and non-linear correlation. In linear correlation, when changes in one variable tends to bear a constant ratio to the amount of change or in other variable, that is linear. But when you are talking about non-linear correlation, then the change in one variable does not bear a constant ratio to the amount of change in other related variable. Now it comes to simple, multiple, and partial correlation. When only two variables are studied, then it is simple correlation. When relationship among three or more than three variables are studied, that is multiple correlation. Partial correlation, when relationship between two variables is examined, keeping other variables as constant. So now it comes to what is correlation and linear regression. So the purpose of correlation is description, inferential statistics, and linear regression is prediction, designed experiments. Statistics, here you can see what kind of statistics could be analyzed here. Then it comes variable in case of variables in correlation, paired, while in linear it is 2, 3, or more. Then variables, no differentiation between the variables, while in linear regression, one or more are independent. Then fits in a line through the data. Yes, implicity exists for correlation. Explicity is equal to y equal to a plus bx, that is linear regression, linear equation you are using. Cause and effect, it does not address any such things, while in linear regression, they attempt to show the cause and effect. Now, we are showing the correlation with an outlier, while correlation after removing the outlier would look like this. So these two are representation of the correlation before and after outlier. So if you want to plot the correlation with outlier, the correlation plot looks something different when you are removing the outlier. Now it comes something called Simpson paradox. These are quite simple, but in statistics it is used. So one not uncommon surprise when analyzing data is Simpson's paradox, in which correlation can be misleading when confounding variables are ignored. So next part is coming some different correlation. For example, correlation and causation. So what is correlation and causation? In correlation, variables do not necessarily have a causal link between each other. While in causation, there are causal link between two variables. So difference between correlation and causation are quite clear. In one correlation case, variables are mostly independent, while in causation, the variables are interdependent. And this can be quite easily seen in this drawing where germs are leading to bad smells and germs could lead to disease. That means they are dependent on each other. But bad smells and disease are independent from each other. The smells can be coming from any source. So this is called correlation. And germs lead to bad smells, that is causation. Causation, germs lead to disease, that is causation, because these two things can be arise from germs. So they are dependent. Now, associate some different way of correlation you can see: associated, positively correlated, not associated, completely discrete, associated, increasing variance, negatively correlated. Here you can see. So in positive correlation, you can see going up, negative correlation going down, then not associated is here. Now, in the next part, I will be covering the probability. So you have seen the statistic parts. Now it comes probability, which will be more interesting when you are trying to calculate the p-values or significance of some kind of occurrences. Then it comes the probability value, which is normally we call it less than equal to 0.05 or 5% probability. These things are called quite significant things in any kind of big data analysis. And thank you very much. I hope that you will be enjoying this